Hello everyone, viewers of Biotechnica subscribers and all CSIR enthusiasts. Today I am going to discuss one more series of Unit 5 important previous year question papers with solution, and today's topic is Urchin Development. This particular topic is not mentioned in syllabus, however we do see questions from it considering it as a general organism in which development can be studied. Processes like gastrulation, cell fate development, axis formation, and a little bit of regulation. So let's see which type of questions come and what are the explanations for them. The first question from CSIR question paper June 2021. This is based on the cell fate of the sea urchin and the different cells of the blastula, that is after the cleavage formation. What different cells are present, you have to know their names, and what is the role of beta-catenin will be helping you to solve this question properly. During normal development of sea urchin, beta-catenin accumulates predominantly in the micromeres, which are fated to become endoderm and mesoderm. If GSK3, it is a molecule in the beta-catenin pathway, if it is blocked in the developing embryo, then what out of these will be the result? So which of the following statement is correct? You have to answer this part. In order to understand this question, first we need to see the beta-catenin pathway and what is the role in the specifying vegetal cells. Now when it comes to the sea urchin, this is what a sea urchin early embryo stage looks like. So this is a 16 cell stage in which there are 3 categories of cells. The ones which are micromeres, they are generally present in the vegetal pole. We know the bottom side, the vegetal pole, where opposite of the nucleus, wherever the nucleus is present, the opposite part of that egg is called as the embryo, is called as the vegetal side. So micromeres, they are present towards the vegetal side, which is followed by a series of macromeres, and on top of that macromeres is a layer of mesomeres. You can ignore the transplanted micromeres, it is part of the question experiment. Now these micromeres are actually very important in specifying the cell fate of the nearby cells, especially for the macromeres. And micromeres have an autonomous mode of specification. That means they can get specified on their own what cell types they are supposed to form. And micromeres give rise to the skeletogenic mesenchymal cells, which ultimately form the spicules in the adult sea urchin. So before going into the beta-catenin pathway, I should tell you that this micromeres express beta-catenin in a lot of amount in these vegetal cells. So out of all the cells in the sea urchin embryo, even in the blastula stage also, it is only the vegetal pole where we will see the expression of beta-catenin, and beta-catenin is what responsible for the autonomous specification of the micromeres. So here is the role of beta-catenin in specifying vegetal cells. Micromeres undergo autonomous specification to become skeletogenic mesenchyme, and these micromeres, they produce the initial signals that specify the other tiers of cell. Tiers means the other layers of cell. So micromeres will give the initial signals. The molecule responsible for specifying micromere is beta-catenin, which accumulates in the nucleus of those cells which are fated to become endoderm and mesoderm. From the cells which lie above the micromeres, that is the macromeres, from those cells we will be getting endoderm and mesoderm. Okay, and they will be having some amount of beta-catenin. However, it will be more in the micromeres, but macromeres also will see some amount of beta-catenin. And as I mentioned, the accumulation is autonomous and it is responsible for specifying the vegetal half of the cell. Okay, and this during oogenesis, why the beta-catenin accumulates only the vegetal part? Because when this egg is getting synthesized, that is the process of oogenesis, DSH molecule becomes located in the vegetal cortex of the egg. At the vegetal side, in the cortical part, DSH gets accumulated, and because of DSH only, beta-catenin also gets accumulated in vegetal section only. That's why they appear in the micromeres and they are responsible for autonomous specification. How this beta-catenin pathway gets activated? It is by the WNT pathway, and basically beta-catenin is a transcription factor. Only its presence in the micromere is not enough. The beta-catenin, in order to have a genetic expression, in order to influence the genetic expression in the nucleus, it has to enter inside the nucleus, then only it can regulate the transcription, right? So the activation of the beta-catenin is by the WNT signaling pathway. So we have the WNT molecule, which is the ligand, and it binds with the receptor, which is called as frizzled. It is present on the membrane. Now frizzled is associated with another molecule, which is disheveled, or in short it is also called as DSH, right? Then DSH will be, if it is activated, only when WNT will come and bind to frizzled receptor, disheveled, or in short DSH, it gets activated. And generally in the cell there is a complex which is formed by the molecules GSK3, APC, axin, and these all complex will also bind or attenuate beta-catenin along with it, and eventually lead to its degradation by ubiquitination. But when DSH is activated, it inhibits this complex formation, and because this complex formation is inhibited, beta-catenin also cannot be fixed in one place. This becomes free. Only when beta-catenin becomes free, it enters the nucleus and it acts as a transcription factor there by interaction with molecules like LEF and TCF, which after that will influence the transcription of the necessary genes for specifying the cell fate. This part also I will come to after a certain while. Now this is some experiments which are done, like if in the normal development, this is a 60 cell stage sea urchin embryo, it will be able to give rise to a normal Plutus larva. This is one particular developmental stage, after which the adult formation is done. But if we take only the animal half, so animal half is fated to become only the ectoderm, but that will only be when possible when the rest of the cells are there, so that by cell signaling the rest of the endoderm and mesoderm can be formed. But only if there is ectoderm, then this embryo will not be able to form any endoderm or mesoderm. It leads to complete animalization, that is only ectodermal derivatives are formed. But if with this animal half we can keep some micromeres, then these micromeres are so significant in the formation of the adult that they can redirect this animal part to make a recognizable, more or less recognizable larva as the normal one. Okay, just the difference is that here the mesoderm and endoderm derivatives are actually from the endoderm derivatives are actually from the micromeres only. Okay, they only redirect the cells to become the endoderm. So there is no separate endoderm over here. Now this question asks that if GSK3 is blocked in the developing embryo, then which out of those following will happen? So when GSK3, as we know, it will be inhibiting the beta-catenin, and the WNT signaling pathway is to block GSK only. That is the entire signaling event of WNT pathway, to block the GSK so that they cannot inhibit the GSK, so that they cannot block the beta-catenin. But if without signaling itself GSK is blocked, okay, then whether signaling is there or not, everywhere beta-catenin will be free to enter the nucleus. Okay, so generally we know that beta-catenin will be free only in the vegetal cells. In the endoderm, WNT signaling is not there. Ectodermal layer, WNT signaling will not be there, because of which the GSK3 will be blocked, GSK3 will be present, complex will be formed, and that is why beta-catenin will will be inhibited, right? It won't be able to enter the nucleus. That's why this cannot form into any cells. It will form the ectodermal cell. But if GSK is blocked in the developing embryo, even ectoderms also start expressing beta-catenin. So basically entire embryo starts expressing beta-catenin, okay? And wherever beta-catenin is there, it will only form endoderm and mesoderm. So there is no ectoderm for this embryo, okay? Only endoderm and mesoderm will be formed, no ectoderm will be formed for this, right? So beta-catenin accumulation in the nucleus of large micromere will be inhibited, leading to formation of ectodermal ball. First of all, if GSK3 is blocked, nowhere beta-catenin accumulation will be inhibited. Everywhere beta-catenin will be there, okay? So beta-catenin, this is wrong. Second, beta-catenin will accumulate in the nuclei of all blastula cells, leading to an ectodermal ball. Now all blastula cells is correct, but it won't lead to ectoderm ball. It will lead to endoderm and mesodermal specification, okay? So this is not correct. But third one, as you can see, beta-catenin will accumulate in the nuclei of all blastula cell, because of which all animal cells will get specified as endoderm and mesoderm. So here third answer is the correct one. If you check fourth statement, beta-catenin which accumulate in the nuclei of large micromere will be inhibited, leading to animal cells getting specified as endoderm and mesoderm. Okay, so it doesn't accumulate only in large micromere, it will accumulate in the entire cell, right? So that was the answer for that question. Coming to the second question, which is on a similar note. So this is from June 2018. Given below are a few statements regarding the role disheveled and beta-catenin in the development of sea urchin. DSH is localized in the vegetal cortex of the oocyte before fertilization and in the region of the 16-cell embryo about to become the micromeres. Or same similar statement that DSH is localized in the cytosol. So whether it is located in vegetal cortex or whether it is located in cytosol. So this one I have already mentioned to you that DSH is localized in the vegetal cortex. So here A will be the correct option. Then C statement says beta-catenin accumulates predominantly in the micromeres and somewhat in the veg2 layer of cells. So when you look at a sea urchin embryo, the bottommost layer of cells are the vegetal cells, that is the micromeres are the cells. After that you have the macromeres, which are specified into veg1 layer and then again veg2 layer, and top one you have the mesomeres, okay? So they will be forming the animal layer of cells, that is AN1 and AN2. So this is the layer of cells which are formed. As we know that yes, beta-catenin predominantly we find in micromeres and somewhat we will also find it in the veg2 layer of cells only, okay, not in the veg1 layer. So even C is correct. D states treatment of embryos with lithium chloride does not allow the accumulation of beta-catenin in the nuclei of all blastula cells, and the animal cells thus become specified as endoderm and mesoderm. So for this we have to see a certain thing, then only you will understand. So here correct answer is E. Here basically during normal C development, beta-catenin accumulates in the nuclei of those cells which predominantly in the micromere and lesser in the veg2 cell, fated to become endoderm and mesoderm. This accumulation is autonomous and can occur even if the micromere precursors are separated from the rest of the embryo. So this accumulation appears to be responsible for specifying the vegetal half of the cell. So here you can see that the beta-catenin here, it has accumulated in this part, and wherever the beta-catenin is there, that means GSK has been blocked there. That means WNT pathway is there, okay? But when you treat it with lithium chloride, so this is one of the experiment, lithium chloride when you treat, lithium chloride by default it will be blocking the GSK3, okay? As a result, whether signal is there or not, in all the cells GSK3 will be blocked, just like how we saw in the first question, okay, first instance. So there all the cells will be expressing the beta-catenin, means all the cells will have the beta-catenin in its cells. So therefore there will be no ectoderm. Animal cells also become specified as endoderm and mesoderm. cells. Okay, so it will be a completely mesoderm and endoderm. There will be no ectoderm, just like the previous question. Then there is another treatment where you have beta-catenin. The second experiment is you have beta-catenin in cells, in vegetal cells or wherever cells, but you don't allow the beta-catenin to enter into the nucleus. So if you treat it with cadherin, then the nucleus, the beta-catenin transportation to the nucleus will be inhibited. If that is the case, then even if beta-catenin is there, it cannot enter the nucleus, it cannot act as a transcription factor. And that is why even if it is there, it will not be able to specify the cells as mesoderm and endoderm. As a result, the entire embryo results in the formation of ectoderm. That's why it leads to the formation of ciliated ectodermal cells. So what is the statement D saying over here? That treatment of embryos with lithium chloride does not allow accumulation of beta-catenin. This is wrong. It allows accumulation of beta-catenin because treatment of lithium chloride blocks GSK3 in all the blastula cells, in nucleus of all blastula cells. Okay, and the animal cells thus become specified as endoderm and mesoderm. This part is correct, only does not is not correct. That is why D is wrong. Now E states when beta-catenin is prevented from entering the nucleus, the embryo develops as a ciliated ectodermal ball. So obviously if you don't allow beta-catenin, which is a transcription factor, to enter into the nucleus, the embryo will not be able to give rise to mesoderm and endoderm, which is the major, beta-catenin is the major deciding factor for the vegetalization. As a result, entire embryo results in a ciliated ectodermal ball. That is why the correct statement over here is A, C and E. Then another type of question that comes from this area, it is from the double negative gated circuit, which is for the PMAR HESC signaling. Okay, this is from December 2012, but recently also a question has come from the same part, so you can practice more questions on this. Don't think that this is a old question, so now it will not come. That's why I wanted to discuss this with you. Okay, so here they have basically what is the procedure. In the micromeres we saw the accumulation of beta-catenin. Along with that you also have the accumulation of OTX, which leads to the expression of something called as PMAR. Now when PMAR is there, it will inhibit the accumulation of HESC, and HESC is supposed to be an inhibitor of all this vegetalization factors. Okay, so these will all lead to the skeletogenic mesenchyme formation. Okay, but if HESC is there, it will be inhibiting all of this. But if HESC, you can see where PMAR is not there, HESC is active. So HESC is inhibiting all of this. That is why they will not form skeletogenic mesenchyme. They will form the other type of cells. But if PMAR is there, PMAR is inhibiting the HESC, and HESC in turn it will be it will not be able to inhibit the other skeletogenic mesenchymal cells. That's why they will be able to express themselves and they will form skeletogenic mesenchyme. Okay. Now here we have two columns, and in these two columns they have given the injection of certain things, whether mRNA or antisense mRNA, and the developmental outcomes from this. So first one is mRNA of PMAR, which means that PMAR is present. So if PMAR is present, obviously it will inhibit HESC, and if HESC is inhibited, then they will not form skeletogen mesenchyme. Okay, so PMAR mRNA of means if HESC is not there, then the inhibition of these all will not occur, and as a result these skeletogenic mesenchyme will be produced. And for skeletogenic mesenchyme, the cell should start ingressing into the blastocele. So A will be 1. Okay, mRNA of HESC means HESC is present. If HESC is present, then they will be inhibiting the skeletogenic mesenchymal factors, and skeletogenic mesenchyme will not be formed. So B will be 2. Antisense of PMAR means PMAR mRNA, if it is there, antisense mRNA will go and block it, which means PMAR will not be there. If PMAR is not there, HESC will be active and these will be inhibited. As a result, skeleton mesenchyme will not be formed. So C will be 2, and antisense of HESC means HESC mRNA will be blocked. If HESC mRNA is blocked, then these all can't be inhibited. So in that case, skeletogenic mesenchyme will be formed and all cells will be starting ingressing into the blastocele. So that is how you had to match it. So C will be 1, so C will be 1 and D will be 2, and A will be, sorry, C will be 2 and D will be 1, A will be 1 and B will be 2. So option is answer 3. Here is the gene regulatory network which I was just talking about. And this is the double gated negative circuit that we just studied. So remember, in micromeres, activated PMAR represses HESC repressor, and in Veg2 cells PMAR is not activated, because of which HESC gene product shuts down the skeletogenic cells. Okay. This is one more question from June 2021. This is a part C question, experimental one. Try solving this question and let me know in the comment section what would be the answer for this. You can also ask me for the explanation if you are not able to solve. I will make another video for the same. Thank you everyone.